Okay, this is David Zierler, Director of the Caltech Heritage Project. It is Friday, January 7th, 2022. I am so happy to be here with Professor David J. Anderson. David, great to be with you. Thank you so much for joining me today.
Thank you. It's a pleasure to be here.
David, to start, and I know this is going to be a complicated answer, but could you tell me your titles here at Caltech? I say titles because I know you have more than one.
Okay, yes. My favorite title is the Seymour Benzer Professor of Biology. That's a purely honorific title, and then my official titles are the Tianqiao and Chrissy Chen Leadership Chair and the Director of the Tianqiao and Chrissy Chen Institute for Neuroscience at Caltech. And in addition, I'm a Howard Hughes Medical Institute investigator, like Elliot. And so that's a dual appointment. It's a little unusual in that I'm technically an employee of Howard Hughes, although I'm still a professorial faculty member at Caltech.
David, so much to discuss just in the title. Let's start first with Seymour Benzer. It's always interesting to see how these honorifics come about. First, do you see any intellectual connection or heritage between your research and what Benzer did?
Yeah, very, very much so. In fact, I should say that when Caltech, it was actually, I think, David Baltimore conceived the idea of applying honorific titles to Howard Hughes investigators who don't have official named chairs at Caltech because their salary is paid by Howard Hughes. I was originally assigned to be the Roger Sperry Professor of Biology, which was, of course, flattering because Roger Sperry is Caltech's only Nobel laureate in neuroscience. But I had a much deeper connection to Seymour. It was Seymour who personally recruited me to Caltech in the early 1980s, and it was Seymour's example that finally persuaded me to switch mid-career from the study of neural development in rodents to the study of behavior in fruit flies as well as in rodents. And so after Seymour passed away, I asked whether my title could be switched from the Roger Sperry professor to the Seymour Benzer professor. And that was cleared with Seymour's widow, Carol Miller. And since it didn't involve any money or funding, that was fine. But I very, very much feel like I'm a scientist in Seymour's spirit, which is I pursue things that interest me because of my curiosity, not necessarily because I'm trying to cure a particular disease or something like that. And so, for example, Seymour was always fascinated by the fact that, first of all, Seymour was fascinated by sex, and he was fascinated by the fact that copulation in Drosophila typically lasted about 20 minutes, which is the average duration of copulation in humans as measured by Masters and Johnson, as Seymour liked to point out. And so Seymour was very fascinated when he was able to identify a mutant, which he named Stuck, for obvious reasons. The copulation durations were longer than 20 minutes. And although I think he never really figured out what Stuck was, later on in my lab, and we were doing an analogous kind of screen for neurons, we discovered a neuron whose silencing caused a Stuck phenotype as well. And it became clear that this neuron controlled the relationship between the duration of copulation and the timing of ejaculation in Drosophila. Now, this is a topic that, if William Proxmire were still giving the Golden Fleece Award for, researching useless areas of science, I think the control of erection and ejaculation and copulation duration in flies would be at the top of his list. But although Seymour by that time had passed away, I felt that it was my responsibility as sort of one of Seymour's inheritors, even though I was never his student, to pursue this. And so we did, and it turned into a really interesting paper, which has since been followed up by other labs. And it turns out that it's an extremely useful system in which to understand the control of internal motivational states, the duration of behavior, and sensory motor feedback control. So even something that looks as superficially useless as studying the duration of copulation in fruit flies can actually tell us things about nervous system function. So but there was a conscious effort to follow in Seymour's footsteps by studying that problem.
David, we'll certainly return more to Benzer's influence on your research, but moving down the list, as an investigator to the Howard Hughes Medical Institute, does that mean that all of your funding comes from HHMI? Does that free you up from grants that you would have to get otherwise from NSF or NIH or elsewhere?
No and yes. So yes, it provides a cushion of funding that allows me to pursue whatever direction of research strikes my fancy without having to write a grant to NIH or NSF to support the research. But no, it doesn't provide enough funding to support my research effort in the lab. It covers about 40% of the funding. And so it's a mixture of NIH funding and HHMI funding. But it's been extremely useful as basically a source of funds that I can use for any purpose I want.
Now, being a medical institute, does the funding or affiliation with Howard Hughes come with any expectation that some of your work should have a translational or a therapeutic component to it?
No, there's no expectation. I mean, Professor Meyerowitz, who works on plant genetics, is a Howard Hughes Medical Institute professor. And, you know, the applications of plant genetics are primarily in agriculture, not in medicine, although clearly there are important plant natural products that have medical importance. But at the beginning, the HHMI, Howard Hughes Medical Institute, has evolved. It was originally created as a tax dodge by Howard Hughes in the 1950s. He got a bunch of friends of his who were physicians, and he anointed them as the Howard Hughes Medical Institute and gave them money to do I don't know what. And he was able to write off some amount of money from his taxes in this way. And in fact, HHMI was the first MRO, medical research organization, to be recognized by the Internal Revenue Service. So it's not a foundation. It doesn't give grants. What it does is it pays employees to perform scientific research for it. And that's why all HHMI investigators have to be employees of HHMI. We get, even though we're situated at Caltech, we get our paychecks from HHMI, our healthcare benefits from HHMI, our tax deductions are paid by HHMI, and so that relationship lasts until we get terminated or resign from HHMI, because we get renewed every 5 years or every 7 years. And so if a Caltech professor gets terminated from HHMI, and they're tenured, they revert to being a Caltech professor, and then they go back to getting their salary and their healthcare benefits and everything else from Caltech. But the advantage for Caltech is that since I became an HHMI investigator in 1989, Caltech hasn't paid a dime of my salary since 1989, which is 32 of the 35 years that I've been at Caltech. So I think I've been a good deal for Caltech.
David, from a service perspective, being essentially an employee of HHMI, does that affect what kind of committee work, divisional chair work, those kinds of things that you would do if you were a professor?
That's a really good question. In theory, it was supposed to free investigators up from all of that kind of stuff. In practice, that has not happened at Caltech because I think the divisional leadership was always concerned about creating a sort of two-class system of people who have had to do committee work and teaching and other things, and because they had Hughes, and people who didn't. So it has not decreased by in any quantitative way the service that I've had to do for the Institute while investigator. I've been on many committees, divisional committees, institute-wide committees, teaching, but there is a limit, there's a cap that you can't spend more than 25% of your time on non-research related activities if you're an HHMI investigator. So although I'm the director of the Chen Institute, since that only really takes up about 10% of my time or 15% of my time, that's not a bad thing. I doubt that I probably could not be division chair, because that is probably more than a 50% time commitment, and certainly not something like provost. So division chair is sort of on the borderline.
Now, to flip that question around, are there responsibilities you have to HHMI in the service realm beyond simply doing the research that's the nature of your employment with them?
No, not really. I mean, I have to not what you would call service. There are no committees. I mean, occasionally I am asked to review applications for new HHMI investigators, but A, that's not obligatory, and B, I get paid an honorarium to do that. So it's made clear that this is a voluntary thing that is not part of the expectation of your performance and as an HHMI investigator. And when you're evaluated for renewal, it's based exclusively on your publications and your research performance. There's no aspect of service that comes into play.
Now, maybe this is a petty question, but when you receive a major award, and certainly you have, does Caltech and HHMI want both to claim you so that they're sharing in that honor for you?
I have no idea. I don't think any award that I've won is major enough that they would be quibbling about that. I think the one thing where they do contractually share, the thing they contractually share in is intellectual property. Mm-hmm. So technically, any patents that come out of work that I do are owned 50-50 by Hughes and Caltech. And there's some sharing arrangement if there are any royalties on how that works. But Hughes pretty much lets Caltech decide what to do in terms of licensing IP to a startup, for example, and Caltech pretty much lets its professors decide what they want to do.
David, moving on to the Chen Institute. First, were you present at the creation when the Chens were inspired to make this incredible gift to Caltech?
I was present at the signing of the documents, which was an important event that occurred in Singapore in 2016. I was there together with Tom Rosenbaum and Ed Stolper, the provost, and Steve Mayo, who was division chair at the time. But I can't claim any credit at all for attracting the Chens to Caltech or in persuading them to make the very large and generous donation that they did. I think that is largely Steve Mayo's accomplishment, together with people who were in the development office at the time.
David, do you have any insight as to why the Chens were inspired to partner with Caltech of all the institutes in the world?
That is a really good question. And that actually came up at the signing ceremony in Singapore. And they made 2 points that I thought were quite interesting. One was that at the time, one of their advisors had a background in engineering, and Caltech so they were used to dealing with engineers, and they felt comfortable talking to, negotiating with engineers, and I think that and also with the small and compact size of the Caltech administration. You don't have many layers of deans. We don't have, medical schools, law schools, business schools. Basically, the core decision makers were the three people that were in the room. They're the president, the provost, and the division chair. So that attracted them as a simple organization that they could deal with. Secondly, in China, Caltech enjoys a very high reputation, probably higher, sort of on average, than it does in the United States, where people, for example, most people in California, when I tell them, if they're not in science, I tell them I'm at Caltech, they say, oh, is that the same thing as Cal Poly? No cab driver in Massachusetts, in Boston, would ever ask that question about MIT.
Right.
So Caltech has succeeded in keeping a very low profile. And then the third thing that they said, which I found very interesting, is that they were impressed with the fact that Caltech had race-blind admissions.
Mm-hmm. Mm-hmm.
And because I think it was already at that time, 2016, there were a number of lawsuits ongoing at Harvard and other schools about systematic discrimination against Asians in the undergraduate application process. And so as Asians, Tianqiao and Chrissy were impressed by the fact that Caltech accepted people purely on the basis of merit and background, and as a result, there is a high fraction of East Asian and South Asian students in the undergraduate body, and that there was no discrimination. So that also contributed to it. Whether those were and I guess there was a fourth reason, which is that they really wanted to own neuroscience at whatever institution they made the gift. And for better or for worse, most of our other major competitor institutions, Harvard, MIT, Columbia, Stanford, already had received major donations for neuroscience institutes that had the donor's name attached to it. And we had never received any such donation in the past for neuroscience at Caltech. And so this gave the Chens the opportunity to basically brand neuroscience at Caltech with their name. And so, of course, that makes it difficult now for us to raise additional funding in neuroscience from other people. But I think that was the other reason. They saw an opportunity to sort of make neuroscience at Caltech a Chen Institute brand.
David, are you aware if there is anything personal to the Chens that motivated them to fund research in neuroscience specifically?
Yeah. And Tianqiao has been quite open about this in interviews that he's given to publications. So he suffers from panic disorder. And in fact, at one point, he was the richest man in China, I think when he was in his early 30s, and he made his money from online gaming. And at some point, because of his panic and anxiety, he just decided to withdraw from business altogether and to focus on philanthropy and investing. But he's very interested in trying to develop new treatments for mental illness. And it's still very difficult for him to get on a plane and fly anywhere because of his panic disorder. So I think that's one of the things that has motivated him, number one. The second is that he's an entrepreneur, and he sees what he calls the fourth industrial revolution, which is brain-machine interfaces and the ability to integrate control of human thought, consciousness, disease, treatment, with software and hardware. And in fact, to give credit where credit is due, the Chens were attracted to Caltech by the work of Richard Anderson, who's not related to me, S-E-N, not S-O-N. And Richard has received a lot of publicity. They saw him in a BBC television special for this spectacular brain machine interface work where he works with patients who are paralyzed from the neck down, quadriplegics, and he implants electrodes in their brains that allow him to decode motion intention signals. And so he records from part of the brain that's involved in motor planning. And so basically, if the patient he asks the patient to imagine, say, reaching for a cup of coffee as opposed to doing something else, he can train a computer algorithm to recognize the pattern of neural activity that is associated with the patient's imagining performing a particular action like reaching for a coffee cup. And he can then route that signal, that decoding, to a robotic arm, and control it to reach a coffee cup. And then the patient, using biofeedback over many months, can actually train themselves to control this robotic arm by thinking about what they're doing and then improving the performance of this decoder. And so this has allowed patients who basically can't do anything from the neck down to perform simple tasks like that. Now, all of this is at very, very early stages of development, and it's nowhere close to something that is going to be deployed routinely in medicine, at least not for probably another decade or so, because it involves very, very cumbersome computer apparatus, robotics. Nevertheless, the Chen's imagination was captured by this technology, and they also see this as a future area for entrepreneurship. And so they've set up a number of institutes in China to try to expand the testing and development of this type of technology. And so for them, the support of neuroscience has been tightly connected with the idea of supporting the development not only of fundamental science, but of intellectual property that could then be used and licensed by them if they chose to start companies that develop this sort of technology.
David, given the importance of Tianqiao's panic attacks, did that inform at a foundational level what aspects of the Institute would be directed towards basic or fundamental science, and what would be informed or motivated by translational medicine?
No. And in fact, as the director at the outset of the founding of the Institute, I was actually quite concerned that I would need to somehow protect my faculty colleagues from pressure, whether implicit or explicit, from the founders to direct their research in particular directions. And I'm happy to say that no such pressure was ever exerted or materialized, and that I think the Chens and Caltech now have established a very good working relationship of mutual trust, and they trust us to choose the science that we find most interesting to do. I mean, the one way in which it did in fact affect the distribution of resources in the Institute is that there was a special center that was set up for Richard Anderson to study brain-machine interfaces. So there's three centers within the Chen Institute. There's the Center for Systems Neuroscience, which I'm the acting director of. Doris Chow was the director of that before she left and moved to Berkeley. There's the Social and Decision Neuroscience Center, which is directed by Colin Kammerer, who's in the Division of Humanities and Social Sciences. He's an economist. And then there's the Brain Machine Interface Center, which is directed by Richard Anderson. And so I think those decisions of how to distribute the resources were made before I was recruited to be the director of the Institute. So I played no role in that. Those were decisions made by the president, the provost, and the division chair. But presumably the hefty investment in brain-machine interface was in recognition of the fact that it was Richard Anderson's work that first brought Caltech to the attention of the Chens.
David, to go back to the idea that there's this mutual trust that's been built up between the Chens and Caltech, is there also a general understanding that the Chen Institute is working in an area of science that's really pushing the frontiers, and just by a matter of fact, there needs to be the basic science, the fundamental research, before anyone can even think about translations?
Yes, of course, they totally get that. They have totally bought into that, and I think they have evolved a broader view now of what I've called the sort of Chen Institute ecosystem. So they have their own Chen Institute, which is the sort of umbrella organization in whose ecosystem the Chen Institute for Neuroscience at Caltech is one component and one node. And so they realize that if they want to be able to control the direction of research, to channel it into the development of intellectual property that they can license in order to found startups to develop the technology, that the university is not the right forum to do that. And so they're set, at least not in the United States, they're setting up other institutes and other models. They have one in China that they've set up, which is more set up along those lines, kind of like a Howard Hughes Medical Institute, where the people that are working in that institute are employees of the Chens, the Chen Institute, and then the Chen Institute will own any intellectual property that is generated and be able to license it.
David, to go back to your titles on a day-to-day, you're leadership chair, but you're also director of the Institute. What are the distinctions there?
Oh, the leadership chair means that there is some actual funds that are given as part of the endowment distribution to whoever is the current Tianqiao and Chrissy Chen leadership chair. So when I step down from being director, that will go to somebody else. And those funds, they don't fund my laboratory. They go to largely to supporting the salary of my executive director, Mary Sakura, who is the person who's excellent and makes everything happen in the Institute, and also a part-time administrative assistant. And then any funds in that are left over go to support other activities that the Chen Institute sponsors around campus. And I can go through those, but those are the sorts of things that I do as the director of the Institute.
Well, David, a solid half-hour discussion just on all of your titles and responsibilities. No, it's incredible.
Not very interesting.
No, it's wonderful. It's great insight into all of your responsibilities. Now let's turn more to the research. So first, to take a wide-angle view of the course, the trajectory of your career, and how it changed as a result of Seymour Benzer's influence, can we also say that Benzer himself became involved in emotion, in this kind of neural circuitry that was not part of his earlier career?
Only to the extent that he was a co-author and a reluctant co-author at that. I can tell you that story at some point later on.
Yeah.
On our first paper on fly behavior that got into the area of emotion, what looked like quote unquote fear in fruit flies, and it was based on a anecdotal observation that Seymour had made that we decided to follow up on. And Seymour was a co-author with me and my postdoctoral mentor, Richard Axel from Columbia, on that first paper, which was published in Nature in 2004. In fact, I think it was published just before Richard received the Nobel Prize for his work on olfactory receptors. But Seymour was not really interested in that particular topic. He was interested at that point in his career in neurodegeneration, and did a lot of work on that. He'd been interested for a long time in the question of neurospecificity, that is, how brains get wired together, how all the neurons know how to find their targets. And then the reason that he got into the fly in the first place is that he wanted to understand the relationship between genes and behavior. And so he was studying innate behaviors like courtship behavior or geotaxis behavior, simple behaviors, although courtship is anything but simple. But he never really identified emotion as a topic that he wanted to study in flies. I guess it was something he took for granted. You know, most people, if you ask them, do you think that flies have fear? They would sort of shrug and say, well, I kind of doubt it. They're probably just little robots. But as far as Seymour was concerned, it was a foregone conclusion. He said, of course flies have fear. You put them in certain situations, they have to crawl through little holes. He said they don't like to do that. So Seymour endlessly anthropomorphized his flies, as it's very tempting to do that when you study them for a long time. They start looking like little people with more legs than we have and with wings. But that really wasn't the area that he was interested in.
David, for you, I wonder if you can explain first, just at a very general level, what neural crest stem cells are.
Sure. So for the first half of my career, basically from the time I got to Caltech in 1986 until 2005, I studied the development of the nervous system and focusing on how different types of cells are generated during the development of the nervous system, which is, of course, it's a sub-problem of the most fundamental question, which is how does a single fertilized egg, a zygote, develop into an entire organism that has, hundreds and hundreds or many tens of thousands of different types of cells and different tissues and different organs, not just the different cell types in the brain. And the neural crest is a part of the developing embryonic nervous system, which sits atop the developing spinal cord. So the developing spinal cord is a longitudinal tube in a fetus, which is actually called the neural tube. It rolls up from a sheet, an elongated sheet, to form a tube, and that tube will give rise to the central nervous system, including all the neurons in the spinal cord and all of the neurons in the brain. It sort of balloons out into a bunch of vesicles at the front end that become your cerebrum, your cerebellum, etc. But there's also an important part of your nervous system that is outside the brain and the spinal cord, which is called the peripheral nervous system. And that consists of sympathetic neurons and parasympathetic neurons that control things like heart rate, respiration, blood pressure, so-called autonomic functions, and also your enteric nervous system, the neurons that control your gut and digestion, which have become a very hot topic recently because of all the interest in the gut-brain axis. And all of these peripheral neurons come from this precursor cell population that forms, it delaminates from the top of this rolled up neural tube, and the cells migrate away from their site of origin on top of the neural tube, and they spread out through the developing fetus and home to specific locations and become different types of cells in different locations. And as somebody that was very interested in these types of differentiation processes, coming from the work that I did in Richard Axel's lab as a postdoc, I wanted to understand whether those decisions, those developmental processes, were generated in some sort of a hierarchical pattern with some sort of Ur cell at the top that was completely undifferentiated, then sort of in a hierarchical way generating progeny that became more and more and more specialized, or whether to begin with the neural crest itself was very heterogeneous, even though it looked in the microscope very homogeneous, and that there were predefined subsets of cells that were going to generate different types of derivatives from the neural crest. So a parallel rather than a hierarchical mechanism of development. Of course, everything is hierarchical when you take it back to the level of the egg, because there's just one cell to begin with. So I was very influenced by another Caltech colleague who's also subsequently passed away, named Paul Patterson. He was a neuroscientist at Caltech, and Paul was very interested in parallels between the nervous system and the hematopoietic system, the system that gives rise to various blood cell types. And he focused on similarities in intracellular signaling molecules that were shared by these two different systems. And there are many similarities, but I became fascinated by the question of whether the concept of a stem cell, which was deeply ingrained in and really evolved from the study of the hematopoietic system, that is the idea of an Ur cell that sits at the top of this hierarchy that does two things. It spins off progeny cells with more restricted developmental options, but it also makes additional copies of itself. For example, by dividing asymmetrically to produce one daughter, which is another stem cell. So you always have a reserve of stem cells, and then a second daughter that starts to differentiate. And I was interested and fascinated by the question of whether the nervous system might develop according to similar rules. And I thought that the neural crest might be an easier system to tackle that problem in than the central nervous system, because the central nervous system is very complicated, has many, many different cell types, and the neural crest seemed like a more manageable system in which one could study that. And indeed, many laboratories before me, like Nicolas Douarin in France and Marianne Bronner, who's my colleague here at Caltech, have done lots of work mapping out the fate of various neural crest cells. And Marianne has now done much more work on molecular mechanisms. But basically, a neural crest stem cell was something that we hypothesized might exist in a speculative review article that I wrote in, I think, 1988 or 1990. It was called The Neural Crest Cell Lineage Problem, neuropoiesis. And so I coined or borrowed this term from hematopoiesis, which is the process that's used to describe how the hematopoietic stem cell gives rise to white blood cells, red blood cells, B cells, T cells, macrophages, granulocytes. and raised the question of whether a similar mechanism and a similar kind of cell might exist, at least in the peripheral nervous system. And so then we set out to search for such a cell. And finally, in 1992, we were able to provide evidence, at least from studies in petri dishes, that the neural crest did contain cells that had this dual property. They could differentiate into specialized cell types, neurons and glial cells, which are the non-neuronal cells of the nervous system. But importantly, they could also self-renew. They could make more copies of cells that retained that multipotency, the ability to differentiate into different derivatives. And this was actually the first example of a stem cell that was described in any part of a vertebrate nervous system, and it came right at the beginning of a field that then mushroomed into this area of stem cell biology in the brain and the central nervous system, et cetera. So there have been lots of other people that have worked on neural crest stem cells, and beautiful work by Marianne and others that have confirmed that these stem cells do exist, at least transiently in vivo. What's not clear yet is how long they persist. Do they actually last into adulthood, like blood-forming stem cells do and skin-forming stem cells do, or are they there just for a while and then eventually they peter out? And I don't know the answer to that question, but people are probably working on that. But anyway, that's the long answer to what is a neural crest stem cell.
David, you're talking about neural crest stem cells somewhat divorced from the animals in which they exist. So what are those animals that you were studying? And then I wonder if you can talk a little bit about the ability to extrapolate from one given species to all animal species or not.
Sure. So I should have said that the neural crest it's a vertebrate invention. Okay, it is not something that you find in fruit flies, for example, or in mollusks or other invertebrates, but it's pretty much found in all vertebrate species. And a lot of the early work on neural crest development that was done in avian embryos, quail embryos, and chick embryos. We focused our efforts on mouse and rat because we wanted to bring genetic tools to bear. We wanted to understand ultimately the genes that control these developmental decisions, if you can call them decisions, by neural crest cells. And mice were a better system for using genetics to study that problem than at the time than chick embryos were. But that has since changed. And so the things that we and Marianne and others have found apply to rats, mice, chicks. Marianne does a lot of her work now in zebrafish embryos, which are another vertebrate, and they almost certainly apply to humans as well. And in fact, in humans, there are a number of disorders that are due to defects of various kinds in neural crest derivatives. And so craniofacial malformations such as cleft palate are due to defects in the neural crest cells that give rise to the bones of the face. So neural crest cells are interesting because they don't only give rise to neural derivatives. They also give rise to non-neural tissue like the bones of the face, cartilage, and also outflow tracts from the heart. There are also a number of childhood tumors, neuroblastoma tumors, that come from neural crest derivatives. And then there are various kinds of neurological disorders like Guillain-Barré syndrome, which is a demyelinating disorder that affect neural crest derivatives. So they're not only do humans have neural crest cells, but neural crest cells are their derivatives are the cellular targets of a number of different kinds of diseases developmental disorders, cancers, and neurological disorders that affect humans.
David, obviously it wouldn't be part of your research, but have you seen other researchers, even doctors, take what you found and have applied it in therapies or translational research?
I'm trying to think about this. Not yet. So I actually co-founded a company in 1994 with Irving Weissman from Stanford. He was the person that I isolated, the first person to purify the hematopoietic neural stem cell, and with Fred Gage, who's now the president of the Salk Institute, who worked on brain stem cells to try to see if we could develop therapies based on stem cell technology. And I have to say that I was so we had a lot of patents as a result of the work that we did on the neural crest stem cells that we licensed to this company. And I was very frustrated and disappointed that nothing ever came out of this because although I felt the neural crest disorders of the neural crest, like demyelinating disorders, were some of the lowest hanging fruit in where one could try to get proof of principle that a stem cell therapy could actually work. They were simply not large enough markets monetarily for a biotech company to be interested. So what biotech companies are interested in, what venture capitalists are interested in, are disorders that affect lots and lots of people, like Parkinson's disease, Alzheimer's disease, and these are all disorders of the central nervous system, which the neural crest has nothing to do with. And so whatever focus there was on neural stem cells writ large in that company went into a focus on central nervous system, brain stem cells, and spinal cord stem cells. So I think, sadly, there is yet no clear application of neural stem cell technology. But the fact is, there are very few applications of any stem cell technology that have been FDA approved and are regularly used in the clinic, even for hematopoietic stem cells, which have been around a lot longer than neural crest cells. And hematopoietic stem cells were pushed by Irv Weissman as a better and purer substitute for bone marrow transplants as a treatment, not for a disease, but a treatment for a side effect of a treatment for disease. Namely, patients who get radiation therapy for cancer have their immune systems wiped out because the immune system is sensitive to the radiation. And so typically that has to be replaced by bone marrow transplants. But if the cancer is in the hematopoietic system, if it's a lymphoma or some kind of hematopoietic cancer, you obviously can't transplant the patient's bone. You can't take a first-degree relative and transplant it because they could give you the same mutation all over again. And so the idea is that if you purify the hematopoietic stem cell, you can get rid of any contaminating cancer cells, and you could use the patient's own hematopoietic stem cells. You wouldn't need a first-degree relative, and you could transplant that stem cell to replenish their bone marrow hematopoietic cells. And there have been promising clinical trials, but it's not an approach that is widely in use, I think, in the clinic, to be honest, because it's very cumbersome and very expensive and requires a lot of machinery, and bone marrow transplants are just easier and cheaper to do. And the only other stem cell technology that's showing any promise right now, and it's still very early days, is the transplantation of stem cells to make pancreatic islets to treat diabetes. And in fact, you may have seen just in the last 2 or 3 weeks an article that one of the first human patients to receive such a transplant, this is an N of one, so it's an anecdote, it's not data, seems to be showing dramatic improvements in their diabetes. And this is the work of Doug Melton, a Harvard professor, who started working on stem cells 30 years ago, just like 5 years after I started working on them. So you can see the incredibly slow trajectory that it takes to get from the basic biology of stem cells into something that the FDA would actually let you put in a person and begin to conduct clinical trials.
David, I wonder if you
understand how slow it is.
I wonder if you see the relative lack of progress in the regulatory framework with stem cells as having a political dimension to it, given the controversies surrounding stem cells.
Oh, now you're bringing up all kinds of history that I prefer to avoid. But in the mid 90s, I guess. So stem cell biology has always been, as you imply correctly, a political hot button issue because some stem cell research, human stem cell research, uses tissue from aborted human fetuses. And there are religious and political objections to that on the right. And I actually wound up making a trip to Washington, D.C. With a group of Hollywood film industry, wealthy producers who had children who were afflicted by juvenile onset diabetes, for which stem cell therapy was a potentially important treatment, to lobby various. At the time, there was such a thing as a moderate Republican. And so we went through Capitol Hill. This was quite an eye-opening experience to me because I realized how much money and recognition as these Hollywood industry types have open up the corridors of power to you. So, we met with Orrin Hatch and Susan Collins and other moderate Republicans to try to persuade them of the importance of human stem cell research. And so for a while, I was very involved in that. And then that led to this passing of this proposition in California that what did it do? It mandated $3 billion in state spending for stem cell research over a 10-year period. And that led to CIRM, the California Institute for Regenerative Medicine. It led to Irv Weissman and Rusty Gage establishing major new centers and institutes for stem cell research. And that was precisely the time that I decided to get out of stem cells and to move into a different field. When the money started pouring in. I remember during one of my many sleepless nights at 3 a.m, looking at myself in the mirror thinking, are you out of your mind that you're getting out of this field just at the time that resources and thousands of people are pouring into it? But this was Seymour's influence again. Mm-hmm. So Seymour made one of his first, what you would arguably say, of many Nobel-worthy discoveries, although he never got a Nobel Prize, when he was working on phage, bacteriophage, to help crack the genetic code. And he made some absolutely seminal contributions to that. And then just at the time when the field of molecular biology and the chance to crack the genetic code and establish the central dogma opened up and people came flooding into it, Seymour said, that's enough for me. I'm going off to open up some new area, because Seymour just didn't like to work in a heavily competitive field, and neither do I. And so you're always faced, when you start something new in science, which then brings a lot of people into it, you have to decide whether you want to stay in the field and duke it out with all of the competition that you've spawned, or go off and do something else. So in switching from stem cells to behavior, I was very much trying to follow in Seymour's footsteps of doing the non-obvious thing and going into a new area, which now, of course, has itself become highly competitive, as did stem cells. So you can run, but you can't hide, as they say.
David, of course, this now gets us to neural circuits, and just by way of explaining the transition, how different of a field is this? How much of a leap? Are you hitting the textbooks again? How much are you teaching yourself, or is there a natural bridge between the two areas?
Right, that's a really good question. So there is and was a natural bridge in that I was a developmental neurobiologist. I studied the development of the nervous system, and one way you can argue that one way to understand how the brain works is to understand how it's put together. But the promise of that has not really been realized. And so for the most part, it's been a very radical shift in the way I think about science and fields. But we started off by trying to actually make that link between development, neural crest development, and function by taking advantage of the fact that the neural crest gives rise to neurons that sense painful stimuli. And we used that to bootstrap ourselves into identifying genes that marked very specific populations of pain-sensing neurons in adult animals, and to begin to start to use those genetic tools to map the connections and the circuitry that those neurons were involved in. So in that little part of our work, which was at the very beginning of our transition from neural development into behavior and neural circuits, we did leverage our work on the development of the peripheral nervous system and started out working on pain that is noxious and also pleasant stimulation to the skin and the body. And that actually led to an interesting discovery, which was the discovery of a very rare subset of neurons, this is in mice, that send their fibers to the skin, but instead of responding to painful stimuli like a pinprick or the touch of a match, they respond to pleasurable stroking of the skin. And we were able to show that, and so we loosely refer to them as massage sensing neurons. And of course, these play a very important role in nature, since what animal mothers do, invertebrates, they spend an awful lot of time licking and grooming their offspring. And that licking and grooming is in part, we think, detected by these massage sensitive neurons. In fact, we got the cover of Nature for this paper in 2013, and Nature chose the photo. It shows a mother baboon grooming her offspring in that way. And we were able to show that animals find the activation of these neurons a positive experience, because we can train the animals to return to a location where those neurons had been activated. So that said that there was something, their brains detected something positive associated with that. So there really was a through line that started with our neural crest work and took advantage of genes that we discovered in the course of trying to study the development of these neural crest-derived pain-sensing neurons, and took us all the way into adult behavior.
And so just so I understand, kind of a chicken and the egg question, were you interested in neural circuits and this got you to emotion and behavior, or were you interested in emotion and behavior and that got you to neural circuits?
I was interested first in neural circuits and behavior, and I have to say I try to be pragmatic and opportunistic in the research directions that I choose. And I saw this focus on pain and, as it turned out later, pleasure as a way to leverage the investment that we made in working on neurons that detect painful and pleasant skin stimulation to take us into the study of circuits and behavior in the adult. But in parallel to that, I have to say, I was interested from the outset in understanding emotion, fear in particular. Mm-hmm. And our first paper that was really trying to begin to map the brain regions that control various types of fear had nothing to do with our work on neural development or neural crest. That was published in 2003, and it was a study of how the brain processes sounds that are innately aversive to the animal. So you can take a mouse that has never before been exposed to an ultrasonic tone at about 20 kilohertz and play it to them, and they will freeze or they will try to run away from the place where the sound is being generated. And we mapped the brain regions that are activated by those stimuli and tried to begin to understand what determines whether the animal's brain decides to freeze or decides to run in response to that sound. And for that, I really had to go back to graduate school. I mean, I had to do huge amounts of reading. And I have to say that for a long time, the wind was very much in my face instead of at my back. It was, I had achieved a certain amount of prominence in the stem cell field to the extent that it was very difficult for me, for example, to wander around a poster session in a meeting without people approaching me, students, postdocs wanting to pepper me with questions, and I could never read the posters that I wanted to read. And I remember going to my first meeting on the amygdala, brain region implicated in fear, and nobody knew me from a hole in the wall. And it was actually kind of refreshing to be able to wander around these posters like a first-year graduate student and be completely ignored by everybody at the meeting. In fact, I was completely ignored by that field for the first decade and a half that we worked in it, despite the fact that we published some major papers in Nature on the control of fear by the amygdala. So that involved a lot of retooling and learning. And I have to say, I enjoy science the best when I'm learning something new. The learning process for me is really one of the things that drives me. For example, now, fast forwarding to the last 3 to 5 years, my lab has been getting more involved in taking computational approaches to understanding emotion coding in the brain. And I'm not a computational person. I was never very good at math. If you saw my math SAT scores, I would never have been admitted to Caltech. And in fact, I have no business being on the faculty at Caltech. But the fact is, my father was a mathematical physicist. And although I didn't get the mathematics gene, I do have a pretty good intuitive understanding of mathematical abstractions. As long as I keep myself out of the weeds of doing the actual calculations and computations. And by recruiting to my lab some postdoc and some students who were computational neuroscientists, I've learned a tremendous amount about how to think in computational terms about the brain, even if I'm not myself a practitioner of it. And that's been incredibly exciting to be able to develop my thinking in that direction.
David, a question that has philosophical and perhaps even spiritual components. When you're talking about studying emotion, are you strictly a materialist where you think that all of these things have some scientific explanation, in the sense that perhaps a poet or a clergyman would talk about the soul or metaphysical aspects of emotion?
Yes. So yes, I'm a materialist. I'm a strict materialist. However, it's important to clarify that we use the word emotion, for scientific purposes in a different way than it is used colloquially, just as scientists often redefine terminology for research purposes. So colloquially, emotions are used to refer to feelings, which are conscious, subjective experiences. And those can only be studied in humans because in order to know what a subject is feeling, the subject has to be able to describe it in words, and humans are the only animals that are capable of verbal report. And so what we focus on is an aspect of emotion which you can think of, if you think of the overall emotion as an iceberg, the conscious feeling part is the tip of the iceberg above the surface of the water, but there's a huge amount of brain function in the part of the iceberg that's below the surface, which is unconscious, that we know very little about. And in fact, there is even evidence in humans for unconscious emotional reaction. So even in humans, it's been possible for researchers to show that there can be emotional responses that don't necessarily involve feeling states. So the way I think about emotions, the way we operationalize emotions, is that they are internal states of the brain that evolved as a behavioral control mechanism that controls not only movements, but also controls other aspects of what the body does during a particular behavior, like controlling your heart rate, your blood pressure, levels of hormones. All of these things change when an animal is exposed to a threatening stimulus. And I think of feeling is as the brain's subjective perception of its own internal state. And I'm not going to live to see an understanding of that process of the brain's subjective perception of its own internal state. So my belief that that subjective, that conscious part can be explained materially by understanding the brain is purely belief. It's not scientific knowledge. But I don't need to incorporate that belief into the way I study emotion, because I study the part of emotion that does not involve conscious feeling and conscious experience. And we developed other approaches to studying emotion that don't make reference to or require the ability to measure subjective feeling so that we can study it in animals. And that's really important, because if you restrict your study of emotions to humans, we will be forever left with a purely observational and correlational description of the relationship between brain activity and emotion. So you can put someone in a brain scanner, show them a scary movie, ask them if they feel afraid. They can say yes, and then you can see that there may be activity, quote unquote, lighting up in certain parts of the brain, called like the amygdala, for example, or the prefrontal cortex. But that's just a correlation. It's not causation. It doesn't tell you whether the brain activity is causing the subjective feeling of emotion that the subject reports, although that might be the tempting interpretation that most people would draw. It's equally possible that the emotion is causing the brain activity, and the brain activity is a consequence of the emotion. And then a third possibility is that there's something else that you're not even measuring, which is causing in parallel the brain activity and the emotion state, and that there's no direct causal relationship between the brain activity and the emotion state. It's like violent crime is highly correlated with ice cream consumption.
Yeah.
Why? Is that because violence makes people want to eat ice cream after they've murdered somebody? Or that eating ice cream gives people a sugar high and turns them into violent criminals? No, it's because both things increase during hot weather in the summertime. And so they're highly correlated with each other. So if you want to get past correlation and understand the causal relationships between brain activity and emotion or any other brain function, you have to be able to delve inside the brain and perturb brain function by turning particular brain regions or neurons on or off, and asking what are the consequences of those perturbations for the behavior of the animal or the state of the animal. And those interventions cannot be done systematically in humans because they involve opening up the brain and sticking things into the brain and injecting the brain with viruses and gene modifying the brain. And the only way you can do that in a human is if it's medically justified. For example, neurologists stick electrodes into the brain of epileptic patients to identify the seizure focus so that they can cut it out surgically. And so people do recordings from the human brain under those conditions, and even some stimulation experiments, although those are in any case, they are highly constrained and restricted in what you can do. You cannot, if you want to study humans, you cannot choose to systematically stick electrodes all over the brain just to see what happens when you stimulate the brain or to see how those neurons respond to J-random stimuli. It has to be done, has to be medically justified. So, whether you agree with separate ethical frameworks for studying biology in animals and humans or not, right now the ethical framework that we do work in and that governs research funding says we can do interventional experiments in animal brains in a systematic way that is not justified by trying to cure disease, that we cannot do in humans, which means if we want a causal understanding of emotion, we need a way to study emotion in animals. And that's why we've been putting all of this effort into trying to operationalize emotions as internal states and study them as internal brain states that have certain features or properties. And I can get into these features and properties later on. But to be able to recognize those states and the behaviors that express them, to distinguish them from purely automatic reflexive behaviors. And that's key, because just using your intuition to identify instances of emotional expression in an animal can fool you, because you're just anthropomorphizing the animal. You encounter a squirrel in the park and the squirrel freezes. Your natural inclination is to assume that the squirrel's afraid because you'd be afraid if something 200 times bigger than you suddenly appeared in front of you. But you don't know that. It could just be a simple mindless reflex. And so we've had to develop ways of distinguishing whether an animal's behavior is just a reflex or involves some sort of an internal state, so that we can then study how the brain encodes those internal state properties. And that's really the sort of gestalt of the work. And how we got into this, we were talking about computation.
No, you defined emotion in a scientific context.
That's right.
Not feelings.
That's right. And we're trying to see if we can identify certain computational descriptions of the brain that could explain how certain features of emotion are represented by brain networks. And again, that's something I can get into in more detail later on. But the idea is that you should be able to study emotion in animals just like you can study any other brain function in animals: decision making, learning and memory, pain, any other kind, vision, olfaction. All of these processes have been studied profitably in animals, and yet because we experience emotion in our everyday lives, we think we know what emotions are intuitively, and so we think of them only as subjective feelings in the psychological sense. And that has actually caused some psychologists to argue recently that we should not use animals at all to study emotions, because the way they view emotions is purely in this subjective conscious experience way. So there have been a couple of important books that have been published on that, and I have a book coming out in March which is aimed at a lay audience whose focus is to try to rebut that line of argumentation and to describe why it's so vitally important that we develop ways of studying at least features or aspects of emotion that are common to humans and animals that don't depend on being able to objectively measure conscious feeling or subjective experience in an animal.
So ironically enough, what you're saying then, if I understand, is that animal experiments allow us to ask much deeper questions about emotion than if we're strictly limiting ourselves to humans. That would be akin more to like stamp collecting.
That's right. That's right. It's well, I think stamp collecting is a little harsh. Since you've worked on the history of physics, you'll appreciate this. There's really two different epistemological frameworks for understanding the brain, and one of them is based on purely observational experiments, and I analogize that to astronomy. Okay, astronomy is based purely on observations. The laws of planetary motion were based on observations. They were not based on perturbations. You can't remove a planet from the solar system and see how it changes the motion of the other planets. You can't add an extra planet. You can't move a planet closer to or further from the sun or change the mass of a planet, right? Those are the kind of perturbation experiments that biologists do to understand cause and effect and mechanism in living systems. So the epistemology of astronomy is one lens through which people study the brain. And from that perspective, putting people in brain scanners and correlating patterns of brain activity with verbal reports of emotion could be analogized to looking through telescopes at planets and trying to derive laws that explain planetary emotion purely through observation and correlation. But where biology has been successful in understanding disease, cancer, basic mechanisms of development, cell function, is through this causality-based approach that involves loss of function, gain of function manipulations to ask whether certain genes or cells or proteins are necessary for a certain process to occur, sufficient for a process to occur when activated outside of their normal time and place or at higher levels. And that's the whole causal framework through which Elliot Meyerowitz understands the development of plants, and it's the basis for using genetics to understand any kind of biological process. But that's a different epistemology, and if you want to apply that epistemology to studying brain function, that is one in which causality defined in that way is central, you need to be able to do perturbation experiments and you need to be able to work in animals. So I don't want to go as far as saying that, human neuroscience is just stamp collecting. It isn't, but it's an epistemology that is ultimately going to limit our ability to develop new drugs and treatments for psychiatric disorders. And there's a reason that there hasn't been a fundamentally new psychiatric drug in the last 50 years, even though it only took us 1 year to develop a COVID vaccine. And that is that most treatments in other areas of medicine arise from an understanding of what normal process goes wrong in a disorder. Once people learned that diabetes reflects an insufficiency of insulin, and that insulin is a protein that you can purify from pigs and inject into a person, it immediately suggested a treatment for diabetes. Give the diabetic more insulin. There's no psychiatric drug that has been developed through that process of understanding a normal brain function and what goes wrong in a disorder like depression or schizophrenia. Every psychiatric drug that is used, Prozac, whatever, they've all been discovered purely by serendipity, and we have no idea of how those drugs even work to exert their functions. So it's no wonder that that most drug companies have given up on trying to find new psychiatric drugs, because every time they do, they bomb out when they try to do it on purpose instead of by accident. And so my argument is that until we achieve this gets back to your materialism question, until we are able to understand the brain functions that underlie processes like anxiety, fear, and anger in the way that we understand that the pancreas controls sugar levels in the bloodstream by producing the hormone insulin, which binds to a receptor that takes the glucose out of the bloodstream. Until we get to that level of mechanistic understanding, we're never going to be able to develop new drugs for treating psychiatric disorders.
David, last question for today. By definition, the questions that you're after must take an interdisciplinary approach.
Yes.
So either by individual collaborators or fields that are most important to you, physiology, pharmacology, radiology. What are the most important areas of expertise that you need to help get answers to these questions?
Yeah, that's a really good question, because this type of neuroscience is probably the most interdisciplinary aspect of neuroscience. And so within my lab, we employ molecular biology to manipulate genes. We employ optical methods to perturb neuronal activity in intact brains, to measure activity in intact brains. That means that we have to have anatomical methods for identifying the connections in the neurons in the brain region. So molecular biology, neuroanatomy, behavior. We need to be able to measure objectively behavior in the animals whose neurons we're perturbing. We also use computation because we have to be able to analyze these incredibly high dimensional data sets that we generate from measuring animal behavior in frame by frame in videos at 30 frames per second for hours at a time, or to record from hundreds of neurons that are firing with millisecond time resolution. So there's computation, anatomy, behavior, molecular biology, physiology, electrophysiology, and genetics, which is an extremely important component of our approach as well. And so that's sort of I have kind of one or two people that represent each of those approaches in my lab, but I also have collaborations with other investigators at other institutions and also in Caltech to be able to provide the expertise that I don't have. I mean, I'm at a point now, and this is kind of embarrassing to admit, but it's true. I don't know how to do anything that the students in my lab do, except maybe cut sections through the brain and stain them with antibodies and some of the molecular biology that we do. Although even the molecular biology techniques in the last 30 years have progressed past what I was trained to do with them. But the other things, if you gave me $5 million, put me on a deserted island with no students or postdocs and told me to reconstitute my lab in situ, and here's all the money and construction workers you need, I couldn't do it. So it's all because I'm able to recruit people and they can cross-train people so that when they move on, new people come into the lab to do that, and then being able to collaborate with people outside. Like I collaborate with Pietro Perona, who's an electrical engineer at Caltech and who's a specialist in AI, to develop methods for automatically measuring behavior in fruit flies and also in mice, so that my students don't have to sit in front of a computer monitor 8 hours a day, 7 days a week for months to be able to measure all the parameters of the animal's behavior, but we can do that automatically with computer vision and machine learning. And then I collaborate with people who do things like electron microscope connectome reconstruction of all of the synapses in the fly brain, and have tools that they use to analyze those connections, so we can use that information to guide our experiments in tinkering with individual types of neurons in the fly brain. And of course, computational collaborators as well. So it's a 5-ring circus, and it does. You have to you have to understand what your collaborators are doing well enough to ask critical questions, and you have to develop a good intuition and radar for when things don't make sense and there might be something that is wrong, because ultimately I have to take personal responsibility for everything that's in my paper. And even if it's a mistake that's made by a computational collaborator because they made a calculation incorrectly, I have to take responsibility for that. So, there is a certain amount of skating on thin ice here, but that's why I work with collaborators over many, many years that I trust and have a long relationship of publishing. I've been working with Pietro Perona since 2007 or 2008, and we published many, many papers. In fact, we're almost like a scientific married couple at this point. So I think, and that's one of the things that make Caltech such a great place to be, because I can reach out and find people with expertise, particularly in these mathematical areas that I'm not capable of doing myself, but that are really important to try to put together an integrated picture of how the brain works. So, and that's really the ultimate goal here, not to try to understand the brain just at one level of analysis, like at the level of populations of neuronal activity, but by focusing on specific brain regions and behavior, try to vertically integrate across scales, from the scale of genes and proteins to synapses to types of neurons to local connections in microcircuits to larger scale connections across brain regions, all the way to behavior and to social interactions and social behavior. So that's the admittedly very ambitious, but I still think worthy goal that we're working towards, and that's why we need to take such a broad and interdisciplinary approach to the problem.
Well, David, that's a great place to pick up for next time, where we'll develop all of these issues further. That's great. Okay.